Plant tissue culture is the technical procedure for production of plants from explants. Explant means is the plant pot selected from root to aerial shoot system. Every cell of plant belongs to or similar to zygote-like structure. The zygote develop into complete plant in a normal conditions, but plant cells are similar to zygote condition. They are also produced complete plant on the nutrient medium. This process is called totipotency. Based on the totipotency, we can easily produce the new plants from the explants, from the cells of the plants. Mainly, the plant tissue culture in completely in vitro conditions. Able and invention, different types of methods, procedures for the plant tissue culture. So he was called father of tissue culture. Father of tissue culture. After that scientist experiment, so many scientists did their experiments on the plant tissue culture techniques. So what are the plant tissue culture procedures we followed? Here it is. Steps in tissue culture. So first step is preparation of nutrient medium. Second step is sterilization of nutrient medium. Third step is explant, selection of explant. Fourth step is sterilization of explant. And fifth one is inoculation. Sixth one is incubation. And seventh one is the plantation. These are the main steps for the tissue culture procedure in vitro conditions. Now, first we discussed about the preparation of nutrient medium. See children, in normal environment, plant need nutrients that need nutrients collected from the soil, like some gases collected from the environment, like some temperatures, light factors are also mainly helps for the development of plant in a normal environment. So in vitro conditions, we are provided nutrients and artificial environment to plant tissue culture procedure. In that, first we made nutrient medium. Nutrient medium is essential for the plant tissue culture procedure. Generally, in tissue culture, different types of nutrient mediums are available in market, artificial nutrient mediums. From that artificial nutrient medium, the major nutrient medium is MS medium. MS medium means Murashige-Skog medium. Murashige-Skog synthetic medium is available in all markets of botanical laboratories. In MS medium, inorganic nutrients are present. Like inorganic nutrients are nitrogen, phosphorus, potassium, calcium, manganese, zinc, copper, molybdenum, and etc. The macronutrients and micronutrients, plant needed macronutrients and micronutrients are available in the MS medium. That minerals are called inorganic nutrients. Including inorganic nutrients in MS medium, organic nutrients also present. That organic nutrients major source is sucrose. The sucrose is the major source for the organic nutrients. It is also present in the MS medium. Like vitamins, B complex vitamins are more important for the plant growth and cytokinesis divisions. Amino acids, they are the building blocks of the proteins. These are also present in the MS medium. Including these all nutrients, growth regulators also present. That growth regulators mainly play major role for the organogenesis. Organogenesis means we take single cell or mass of cell as a explant is transferred on the nutrient medium. It develop into organogenesis by the growth regulator, adding of growth regulators. Like growth regulators are mainly two. One is auxins and cytokinins. These are the major components of the nutrient medium, MS nutrient medium. See, it is the first step for the preparation of nutrient medium. The nutrient medium, the next step after preparation of nutrient medium, next step is sterilization of nutrient medium. The sterilization of nutrient medium is major step for the tissue culture procedure. See, nutrients are present in the MS medium, that artificially nutrient medium taken in conical flask or beaker or in test tube in a normal environment. See, if any microorganisms are present on the nutrient medium, they are easily contaminated. That nutrient medium easily contaminated. If the contaminated nutrient medium is used or utilized in the tissue culture procedure, the tissue culture procedure gives a bad results. So compulsory before the inoculation, compulsory the nutrient medium is treated with sterilization procedures. By the sterilization procedure, microorganisms are eliminated from the nutrient medium. See, mainly the sterilization of nutrient medium done in autoclave. Before using of autoclave, we used filter papers. They are also called filter sterilization. By the filter sterilization, large microorgans, any other dust particles, they are eliminated by the filter sterilization. After filter sterilization, we used autoclave. It is the one type of machine for using of sterilization. In this autoclave, we used 15 pounds of pressure. At 15 pounds of pressure at 121 degrees Celsius temperature for 15 minutes. We used which type of nutrient medium like MS medium, like other synthetic mediums are kept in autoclave at 15 pounds of pressure at 121 degrees Celsius temperature for 15 minutes. Automatically, if any microorganisms are present on this nutrient medium, they are eliminated, they are died. Now we collect only fresh, healthy nutrient medium for the tissue culture. During the tissue culture procedure, we use different types of glasswares. The glassware are also kept in the autoclave machines. After the cool down of glasswares, they are treated with ethyl alcohol solution. 95% of ethyl alcohol solution. Finally, the glasswares also sterilized. This is the sterilization procedures for the nutrient medium and utilizing glasswares during the tissue culture procedures. The next step is selection of explant. Selection of explant means which part is used as a tissue culture. Used for tissue culture, that plant part is called explant. Culture using plant parts like from shoot to aerial, root to aerial shoot apex. Culture using plant parts like from root to aerial shoot apex. Like from these all parts between root to shoot apex, all parts are used for explant for the tissue culture procedures. That explants cut from the or select from the roots, shoots, leaf, flower, anther, pollen grains, ovule, embryo. These are the main parts used for the selection of explant. If we used root as a explant, that procedure or tissue culture is called root tissue culture. If you used shoot tissue culture. If you used leaf tissue culture. If you used flower tissue culture. From the flower, petals, sepals, like these parts are also used for the tissue culture. Like anthers anther culture, like pollen grain culture, like ovule culture, embryo culture. These parts are used for the tissue culture procedure as a explant. And the next step is sterilization of explant. Sterilization means we selected explant from outer environment. From outer environment, we selected one explant. Explant means the plant pot used for the tissue culture is called explant. That explant is selected from the outer environment. It is automatically contaminated by the different types of microorganisms in outer environment. So that contaminated explants are inoculated on the sterilization nutrient medium. That tissue culture procedure gives a bad results. So we are used compulsory sterilization procedures for the explants. See, explant sterilization mainly beginning with clean with 5% of TPOL for 15 minutes. See, from the root to aerial shoot apex, which part is selected as a explant? That selected plants pots are kept in 15 minutes of TPOL solution, 5% of TPOL solution. Then after they are clean with distilled water or chloride solution. Explants are cleaned with distilled water or chloride solutions. Finally, we used sodium hypochlorite solution for the cleaning of explant or for the sterilization of explant. And finally, the clean with the water, clean with water. Finally, explant is sterilized. Now, next step is inoculation. Inoculation means sterilized explants transfer into sterilized nutrient medium in aseptic conditions. Sterilized explants transfer into sterilized nutrient medium in vitro conditions or in aseptic conditions. Like for the inoculation, we used laminar airflows. Laminar airflows are maintained in vitro or aseptic conditions. They are also called inoculation chambers. In inoculation chamber, UV lights are present, aseptic conditions are present. Automatically, sterilized explant transfer into sterilized nutrient medium with help of laminar airflow with aseptic conditions. After inoculation, incubation. Incubation means after inoculation, the test tubes are kept in the incubation. Not only test tubes, conical flasks, beakers, like different types of glasswares are used for the tissue culture procedures for the inoculation. See, after inoculation, the test tubes are taken, inoculated explant with nutrient medium. Test tubes are kept in the incubation. In incubation, we artificially maintain 25 degrees Celsius temperature and maintain proper light and maintain proper air. After that inoculated explant on the nutrient medium, 3-4 weeks, after 3-4 weeks, it develop into callus. Callus means it is the mass of cells. Mass of cells are developed from the explant. The explant is utilized nutrient medium and it develop into callus, mass of cells. Finally, we add growth hormones or growth regulators to the callus. Auxins like cytokinins. Auxins, cytokinins are added to the growth regulators, added to the explant. It develop into organogenesis. Organogenesis means it develop into root and shoot system. See, after organogenesis, roots and shoots are developed. They are called plantlets. The plantlets are plantation. Next and last step is plantation. See, up to here, up to plantation or up to before plantation, the plantlets are developed in vitro or aseptic condition. They maintain proper environment. The plantlets are directly cannot enter into the normal environment because normal environment is not comfortable for the plantlets. Before the outer environment, The plantlets are kept in the polythene covers or polythene sheds. In polythene sheds, slightly change the temperature, slightly change the light, slightly change the nutrients. After this procedure, the plantlets are comfortable for the outer environment. Finally, the modified or developed plantlets are plantation at outer environment. The plantlets are developed virus-free plants, quality of food producing plants. The plants are identical to the parental plants. These are the benefits of the tissue culture procedure.